I'll show my daughter how to smash it on her nose and make a nose like mold.
I am like scared to graduate to Play-Doh. Like I haven't. I am way too. I'm not. I'm too ADD person. Like I don't have the ability to like sit and like really supervise Jason's play because I'm like doing them all everything. So I'm just afraid to give her Play-Doh because I'm afraid that it'll end up in my dog's ears or like. All three dogs will have Play-Doh like in their ears, up their nose.
It'll be fine.
Smashed and cracked.
All right, so the answer is A. Good job. Looks like we're at 100% there of the 15 people who. Do y'all not have clickers somewhere? Somebody missing some?
We have like 7 on this table.
All right, we're not actually doing that yet. That's supposed to be after this section. All right, so bear with me for all this stuff because it's a little bit complicated, very zoomed in. Before we get into it, though, let's kind of remind ourselves of what's going on at a neuron that nothing's happening at. So first thing to remember, we've got like the dendrite end, right? These dendrites sticking out. Then you've got the cell body, the nucleus, and then a long axon, and then the axon terminal down here. So dendrite. And if you notice, all the neurons y'all drew were better than this one. Yeah, I know. All right. So the inside of the dendrite, is it more negative or positive than outside the cell? Okay, so we got some negative charges in there. It's positive outside. What about the cell body? Negative or positive inside? A little less confident out there. It's also negative. So negative, 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 positive outside. What about the axon itself over here? It's what? It is. What about the axon terminal? It's also negative. So when a neuron's not sending signals, the whole thing is negative. Just want to make sure we're on the same page there. And the way they send signals is by making it positive. So you let positive ions in, and that's going to trigger more positive ions to come in all the way from the dendrite all the way to the end.
Is a neuron ever not sending signals?
Yeah, all the time. So for example, you've got your pain neurons in your fingers. Is anybody in pain in their fingers right now?
Well, I guess I won't mean the pain ones, but I mean like every part of your body feels something all
right.
the time, isn't it? It's not necessarily pain or.
And as you'll see kind of later, you actually have specific neurons for all kinds of sensations. So you've got pressure neurons sending information about pressure. You've got neurons telling you that you're holding your arm like this. You've got neurons telling you whether you're in pain, neurons telling you the temperature around you. But each one of those is a separate neuron.
So even sensory neurons are even divided further into what types.
Exactly. Yeah. Definitely. So yeah, you could have, some neurons in this spot that are sending signals about temperature, but the ones next to it are pain neurons, so they're not sending any signals.
Nervous system is simple.
Yeah, it's totally simple. All right, so first off, a lot of times the book just says action potential, but a lot of people don't know what that word phrase actually means. So all an action potential is the event that changes the inside of the neuron to become positive and then puts it back to being negative. That's what an action potential is. So it goes from unactivated neuron, activated neuron, back to unactivated. So it goes from negative to positive inside and then back to negative. And another vocab word that you'll see a lot, depolarization. Does anybody know what that is? Okay, so that would be when the inside of the cell becomes positive. And what do you think repolarization is? It's back to negative. So an action potential consists of depolarization followed by repolarization. So as you saw last chapter, or last lecture, the dendrite is the receiving end of the neuron. So it receives all the signals. Now depending on what kind of neuron we're talking about, it could be a neurotransmitter is binding to some receptor out here and letting positive ions in. So neurotransmitter comes by. Imagine it's this pin cap, hops over, binds there, and it's going to let these plus ions through. So remember we did all those channels? What kind of channel is that? If something binds to it and it opens. So it's the ligand gated or ligand gated. So that'd be one kind of neuron. Neurotransmitter comes from another neuron, binds to it, opens the gate, positive ions come in. Or maybe it's a sensory neuron, like pressure. So we start out, negative, and then that neuron senses that there's pressure on your fingers, which opens up mechanically gated channels. So it opens the channel there, and now those positive ions, sodium usually, can come in. Same thing with the temperature neuron. That's mechanically gated too. It'll let your positive ions come in there. So that would be the start of our action potential of the dendrite. Put those positive ions back. Obviously, that's not the whole story. That's the start. So you've got those ligand-gated or mechanically gated channels that open in response to neurotransmitters or, mechanical stimulation. That's the start. And then what would come next is the voltage-gated channels open. So you've got voltage-gated channels that open when the inside of the cell gets a certain amount positive, which, if you can tell from our graph, what do you think the number, what kind of membrane potential has them open? So that's about minus 55 there, which means the inside of the cell got 15 more millivolts positive. And so step one, right, we let in a few positive ions. And then step two is those initial ions change the voltage, make it more positive. And now these other voltage-gated things start opening. So they start opening, and more positive ions can come in. And so if you see on that graph now, it very rapidly shot up to plus 30. So it went from minus 55 all the way to plus 30. So it became very, very positive inside from all those sodium ions pouring in.
So when the inside of the cell becomes more positive, that kickstarts the voltage ones to let more in as
Yes,
well?
right. So it's like a two-step process. Step one is let in some sodium ions. And then once it gets positive enough, then those other channels open, the voltage-gated ones.
So the voltage-gated channels won't ever work without either of the channels or a liquid-gated channel?
Unless you get electrocuted or something. So, and if you notice, what do you think this little one here is? This little pink thing, the little hump. Would be the ligand-gated ones. Let me kind of go back here. So for that little bump there, that's if the signal's not strong enough to get to minus 55. So then the voltage-gated ones never open. So to take an example, your pain neurons, for example, maybe they're sensing a tiny bit of pain at the moment, but it's not quite enough pain to actually create a signal. Like only a few receptors open, and they're letting, I don't know, one sodium ion in. It's not enough to get enough positive to get to minus 55. So step 2, step 3, none of that ever happens. Because the sodium ion gets pumped back out. Or, for example, the pressure one. There's probably some pressure on our fingers all the time, but it's not enough pressure to open enough channels to let enough ions in to get to minus 55. So that's what's called like threshold.
So I keep seeing the minus 70, Michael. Is that a standard number, at least if it's found inside of our
Yes, yeah. So your neurons, nearly all of them are about minus 70. That's just how they were evolved to be. And you remember last time they maintained that by pumping ions out and keeping the phosphates and proteins inside.
So if you have someone that has like nerve damage and they can't feel pressure, pain, temperature, anything, with that, it basically changes their threshold.
If you're talking about, was that from like trauma or something, or something they were born with?
Trauma.
So trauma is usually like damage more in like the spinal cord or something. So like basically breaking the axon somewhere. Now there are genetic conditions where these things would be broken, like one of those ion channels or something, perhaps. Or even it's somewhat common to be like just born without one kind of neuron. Like maybe, because there's people who like don't sense any pain, right? Or at least they don't sense certain types of pain. That would be, yeah. Like break her leg or like.
She stuck her head, and how they figured out it happened to her was she stuck her head on a hot stove and didn't scream.
Oh my goodness.
It burned her hand, and that's how it happened when she was little.
That's pretty scary.
So like people that like are born blind so they're born without that type of.
I mean, there's all kinds of reasons to be blind, but
But ultimately what happens is that it's affecting that.
could affect like the vision neurons, or it could have been like some mutation in the brain, so you're like sending signals, but they're not interpreted right. So anyways, so what does reaching membrane potential threshold mean? Discuss in your group. What does that word mean? All right, so who thinks they know? Who's brave? Yeah? Sure. What is it?
It's like the highest you could reach, like plus 30. That's the highest.
Not quite. It is not plus 30.
That's, yeah.
No, it's a good guess. Okay, what's action potential? Getting closer. There you go. So it's that minus 55. Or you could say letting in enough sodium ions to open the voltage gated channels. Be another way to say it. So as we kind of discussed, once you get to threshold, that minus 55, or enough positive ions come in from this first part of the dendrite, those voltage-gated sodium channels open, sodium ions move in, inside becomes positive. See enough, right? Of course, right now we're still at the dendrite, right? So we've got bunch of positive ions in there. We reach threshold. We open up the voltage gated ones, but we haven't sent any signals down here yet. Now step 3 is getting the signal from the dendrite to the axon terminal way down there. And this is pretty simple because all throughout the cell body, the axon terminal, you have more of those voltage-gated sodium channels. So if we've let in all these positive ions here, at this, like right here, do you think we've reached threshold? This little spot? There's definitely positive ions there, right? So that means that sodium channel is going to open, and then this positive ion comes in. And then there's a channel next to it. Do you think that one's reached threshold? It's gotten pretty positive, so it opens, lets in a positive ion. What about right next to that? Are you going to make me go along the whole neuron and do this? So that one opens and goes through. And that just continues all through the cell body, you know, opening up the channels because it gets positive. And it's almost like positive reinforcement or something, right? Because the one right next to it lets in an ion, which then opens the one next to it, which lets in an ion. So that's how it travels down. And this is the fastest way you could transmit signals through your body. Way faster than like putting a hormone into the blood and letting it get pumped around or something. Because as you know, how fast does electricity travel? Pretty fast, right? Faster than sound. Faster than water, for sure, which is what the blood would be. So this electrical signal goes down, opening up, and then when you get to the axon terminal, then you'd most likely be releasing a neurotransmitter from the end. So NT released. We'll talk about that last step a little later. So there's the propagation, just what I was diagramming and talking about. It's just the adjacent regions reaching threshold, which means voltage-gated channels open, Sodium ions move in, and then it just continues itself on down. All right. So obviously, something has to happen to return the neuron back to its regular state, right? So it's going to be ready to send more signals. And so the way that works is voltage-gated potassium channels open. And they're going to let, because if you remember, this is a little confusing, but remember potassium was higher inside the cell. It's one of the things it maintained. Remember that sodium-potassium pump? So potassium ions are very high in there. So we'll put them in green maybe. So the potassium ions are going to start flowing out through their own channels. So potassium. And then as the membrane potential gets closer to negative, the sodium-potassium pump starts pumping the sodiums out and the potassium back in.
What is it? Just because it's positive? Is that what signals that to start?
Yes, so you have voltage-gated potassium channels that open when the membrane potential is positive. So that like plus 30 that we got to. So as soon as you get positive, they open, potassium starts leaving, which is making it more negative, right? Because positive charges are leaving. And then you're also doing the sodium-potassium pump at the same time, because it's basically always active. And I know it's confusing. To me, it would make more sense just send the sodium back out directly, right? But instead, it's more of a two-step process. So let the potassium go out. And the main reason for that is the potassium's at such a different concentration that it goes out very, very, very quickly. So you can get yourself back to negative super fast. And then you can fix your sodium-potassium levels a little bit slower with that pump. It's going to pump the sodium out and the potassium in. Is that on there? So let's watch this all happen a little bit. And then I'll have you correct a paragraph. Let's see.
Must be rapidly conducted over long distances.
Hold on, I'll restart it.
In order for the nervous system to communicate with other cells, action potentials must be rapidly conducted over long distances. The processes that allow the conduction or propagation of an action potential along the entire membrane of an axon are not very different from those that generate one at the trigger zone. A positive feedback cycle is set in motion when a graded potential reaches threshold and initiates the action potential at the trigger zone of the axon.
So trigger zone's basically the dendrite they're talking about. I mean, it's basically the dendrite. But yeah, I guess right at where the axon is, technically. But for our purposes, it's pretty similar.
Depolarization of the first segment of membrane progresses as the sodium gates continue to open and lasts until the charge is completely reversed. When the action potential reaches its peak, the positive nature of the membrane causes potentials to spread to the adjacent segment of the membrane.
So if you notice our little diagram here, ECF, that's like outside the cell. And then down here, cytosol, that'd be inside the cell. And then in green is the sodium channel, and yellow is the potassium. And so at the start, what was moving? So the sodium channels were moving, or the sodium was moving in.
Simultaneously, repolarization of the first segment of membrane begins with the opening of voltage-gated potassium channels and the inactivation of the sodium channels.
So one thing I didn't mention when that membrane thing becomes positive, what happened to the sodium channel? It closed, right? So it's just open for, a couple milliseconds. And then when it detects that it's positive, it's closed. So that now the only thing that's happening is the potassium's leaving.
The graded potential is unable to reach threshold and generate another action potential in the section of the membrane that is refractory. The nervous impulse is conducted along the membrane to the next segment as the graded potential there reaches threshold and self-propagates the action potential again. This cycle, or wave of depolarizations, continues until the impulse reaches the axon terminal.
All right. All right. So the first thing for y'all to do is correct a paragraph about all this business. So give you a few minutes on your own and then let you work in your group. And definitely feel free to use your notes on this one. Feel like you want to glance back at something. So it would either be ligand or mechanical, depending on what kind of neuron we're talking about. Any extras out there for me so I can remind myself where the mistakes are? I don't know who blows their nose so loudly in that bathroom. right. So you take a moment with your group. I found 1, 2, 3, 4 mistakes. Unless I missed one, I'm going to look one more time. It's different sitting right in the front, isn't it? That's why I make people move around. I'm sure if you offered someone $20, they'd trade with you. Shall find 4?
I found 2.
Yeah.
Is this one right? It's 70. Yeah.
I think the way it's written is fine.
You think?
Well, I may not be exact. I mean, but it does say like approaches. So probably when it gets to -5, they probably start closing. But I wouldn't say it's wrong how it's written. I only found 4, but that doesn't mean that there's not a 5th one that I don't remember putting in there.
Does any of the activity count for the cell body that goes from the dendrites to the axon?
I mean, yeah, I mean it does go through the cell body too.
But it's not an important detail.
I mean, I don't know.
Because nothing in here is cell body, it's all dendrite to axon.
Yeah, I mean, I think if I was rewriting, I'd probably add in, like, say, dendrite and cell body. But the cell body can be anywhere along here. It could be next to dendrite, it could be halfway down. So it's a little hard to put it in a paragraph like that.
So for our purposes, it's 1.
Yeah, I tend to just think, dendrite, axon, axon terminal.
Yeah, because isn't there which neuron has the cell body like off to the side and then the dendrites are here then?
Mm-hmm. Right. Yeah. I can't remember the name of it, but yeah, it's a common one. All right, so let's make sure y'all got them. Is there anything wrong in the first sentence? Should say dendrite, not axon terminal. What about the second sentence? Should be positive ions. If you changed it to sodium ions, that'd be right too. Or you could say positive. What about the next sentence? If the movement of ions into the cell changes the membrane potential enough, action potential is generated. That one's good. Voltage-gated channels in the dendrite and the axon then open and allow Na+ ions to flow in. Sounds good. This triggers voltage gated NA channels in adjacent regions of the axon open as well. That's good. And then dendrite the axon, voltage gated potassium channels open and allow potassium ions to flow into the cell. Out of the cell. So this is our repolarization starting. As soon as that wave of depolarization passes, that triggers the potassium channels to open. So positive ions can leave. And then once the membrane gets back to minus 70, voltage-gated potassium channels close again. That's right. And potassium pump pumps sodium ions into the cell and potassium ions into the cell. Sodium's pumped out. Exactly. All right. All right, so before we do our little activity thing, this is in your book somewhere, and it's nice because it shows you what's happening to the voltage or like the membrane potential, and it's color-coded, so it kind of tells you what channels are open or closed at that point, which is kind of helpful. So you know that, at the beginning, before you get to threshold or before anything happens, the sodium channels are closed, potassium channels are closed, and then if you get a stimulus to threshold, then the sodium channels open, and then the potassium channels open in green here, in yellow, and then when you're back to orange, the K plus channels are open, and the sodium channels are resting or waiting. So, anyways, so if you turn over your worksheet there, got your instructions for this crazy thing I made up. I haven't tried before. And I already noticed when I was going through this that we're missing something. Well, no, I have popcorn kernels. But I mean in terms of one of our channels is missing. So on there, if you write somewhere, blue pipe cleaner is a sodium potassium pump. And I'll give you some blue pipe cleaners. And if you also notice, it was blank for the color of Play-Doh, because I didn't know what color of Play-Doh I was going to give your group. So you should have some on your table so you can write down what color you ended up with there. All right, so basically what you're going to do is model one thing at a time. And when we say modeling it, that means stuff needs to be moving, because this is an active process, right? Things are opening, ions are moving. So what we want to see, so you're going to talk about it as a group, figure it out, and then for each of those three things you're doing, you want to make sure that every person in your group takes a turn actually saying what's happening and moving the stuff around. So if a channel's open, I better see a channel opening. If you tell me ions are moving in, you better be moving the ions in. So it's like a movie, basically, you're making. So I'll bring some blue pipe cleaners and popcorn kernels. You're making a movie. And for this to work, your neuron's going to have to take up half the table, because our pipe cleaners are big, our Play-Doh's big, so we need a big neuron, or it won't work probably. There's a couple of those.
I know, but I don't like this one. So
You need a big one.
I'm gonna draw it like huge.
And if you saw what, you only need to draw like dendrite axon terminal.
Yeah.
We don't need Schwann cells. We don't need all that other business.
Like on.
There's a couple blue ones. There's a couple blue ones for y'all. There's some blue ones. Oh, I think you'll have to make your dendrite wider, so you can actually put so you can see the membrane itself. So just take one of those and just make it way wider, so you can actually put stuff. Yeah, make like a big loop or something, so you can actually put channels in it. Right. So same last time for your dendrite. That's probably how it actually looks. But we need to have a dendrite that's just a big membrane, so you can put stuff in it. So see how my dendrite's got yeah. Yeah, just erase one and make it big so you can see what's happening there. You need to make sure you make your dendrite big enough that you can put channels in it.
Okay.
So instead of a line, make a finger.
Just do one. A duty.
Yeah, just make one big finger coming off of it.
Can it be the middle finger?
Do whatever you want. Sweet. It's my beautiful neuron. So I think for this to work, we're going to have to zoom in on one of these and make it like a big finger. So you can actually see the membrane of it. Yeah, just make one of them big. And then for your axon, I wouldn't draw the Schwann cells because we need a big open space to put stuff in, a big tunnel. I bet there's some extra. Oh, I have to give you all popcorn kernels. There's popcorn kernels. Don't eat them. Here you go. Don't eat those. So whatever works, however you think it looks good. I've never done this before, so just be creative. As long as you can open and close it.
Like,
yeah.
you're just trying to
Mhm.
you can see it enough.
Yeah,
That's how
right.
to do it. Okay.
Right.
The yellow one.
Here's some of these. Yeah, you can. I don't really know the best way to make channels out of pipe cleaners, so I'm just gonna let y'all figure it out. As long as you can make them be open and closed somehow.
Is that good?
Yeah, sure.
What binds to liking?
Neurotransmitters.
Just any, they can be multiple.
I think it's your cotton balls.
Oh, right there. My potassium was covering that.
I see. I see. So I guess you might want to fold up your pipe cleaners a little more. I mean, I don't know how that you're going to show that they're open or closed with them like that. Do you know what I mean? Well, right now that looks.
They're all open or what?
Oh, okay. But right now they're all open.
Should we close some and open
Okay,
some?
that makes sense. Okay. Makes sense how you do it. No, it makes sense. I just, I've never done this before, so I didn't know what people would come up with. And if you're getting stuck, look back at that paragraph you just corrected. So that'll tell you what happens.
So these two are in a similar area. It's just one is potassium and one is sodium.
That's correct.
Okay. And then we need popcorn.
I think I gave. Did I not give you all any?
No.
Okay.
Side.
Sorry.
Are those the ones that are inside of?
The popcorn kernels. So I guess you want to start with an unstimulated neuron, right? So where's the sodium at the beginning? Inside or outside?
Outside. It goes inside.
So start with them all outside then.
Stay in your hole.
Little cool channels there. So my first question for your yellow ones here, how are you going to open and close those? Okay.
We're just spacing
I just think about the structure of them, like
laying them
they
out.
might need to change shape a little bit. Well, I'll keep trying, figure it out.
Is that right?
What?
As the sodium goes in the kernels, then the potassium goes out.
It comes out.
Yeah, and the kernels would go in first,
Yeah, these will come out after.
and then that'll come out right after.
Okay, they go in first and then come out. Oh, I thought it was the other way around. They came out.
Well, I mean, it's all a cycle, right? But if you're starting with non-stimulated neuron,
Filtering.
Ligand gated. So these are all in the ligands.
all in the ligands.
Or
What do you mean?
Dendrite.
Yeah, you do have all three in the dendrite.
And then
Whereas in the axon, you've only got the voltage gated ones.
So the yellow and the pink are only they're in the axon and then
The yellow and the pink are in both places, like you have.
Those go inside, right? Because they're negative. Okay.
They're positive.
They're positive. Wait, the potassium is positive?
And I know that's one of the things that keeps it from not being too negative. But there's also all the negative proteins and negative phosphates in there that we're not showing, which makes it negative.
So they're inside and outside? Those potassium ones?
Yeah, but they're mostly inside.
Inside, okay.
So the way you have it would be exactly right. You know, nearly all the sodium's outside and all the potassium's inside from that pump. And it is,
Negative, no, it's positive, keeping it.
but there's so many other negative things inside the cell that it's negative.
That it's negative,
Right.
okay. There's not enough of those positive ones.
Right, to cancel everything else out. Yes.
So I'm just kind of confused with like what you're wanting to like how to explain.
So
There would be no positive going in, right? For it not to reach the threshold.
well, so what I was thinking for that one is some positive comes in, just not enough.
Okay.
So you get like a very tiny stimulus.
So then if it is reached,
So then,
enough came in,
right.
enough positive.
And then what would happen with all of that? So my idea is you look at A, and then you've got your model here, and you're going to move stuff to show A, and then write down what you did. And then move stuff for B.
So we could take away, right, like our voltage.
We'd have to take away our ions, like our neurotransmitters.
Right.
All these little guys, like the gates will stay.
Now, the only thing you're missing are, do you have your potassium somewhere?
What's that?
Potassium ions? Red Play-Doh?
Yeah.
Where do they start? Outside the cell or inside?
Inside.
Right? And then where's your sodium start?
Outside.
Okay. And the neurotransmitters aren't bound to anything. How about your channels? Are they all open or closed?
They're all closed right now.
All right. So how are y'all doing? It's a pretty good start. And then do you have a way that you're going to show when these are open?
Yeah, just open them. Just open them.
Okay. All right. So I guess the only thing I would change is that they open to outside the membrane. See what I mean? Yeah. So that, you know, they're closed and they open, they'll let those ions in.
The cotton balls,
The cotton balls are the transmitter.
so they stick to the purple.
They'll stick to the purple ones and open up. That'd be the first step. Yes. The sodium potassium pumps, they're all over. So you just have two of them, so just put them wherever.
And those let out what
They pump out the potassium and let in the sodium.
Other way around.
They pump in the sodium.
Wait,
No, wait.
I think that's what you said the first time. They're going to pump sodium out and potassium in.
back in.
Right. So they're like the last thing that's going to happen.
And let sodium in.
That's like the last. That's putting it back to where it's.
Right. Because right now they've already done their job, right? Because everything's sort of active. You may need a few more of these though.
I guess, yeah.
Because do you have any voltage gated sodium channels in the dendrite?
Voltage gated.
Wouldn't it be like around there?
So yeah, I'd put, you know, one over here, one there, one in the axon.
We need a pink one because it goes like his.
Mm-hmm.
It goes like this.
Yeah, because the pink ones are in the dendrite and the axon.
So they're kind of like all over.
Yeah,
Except for that
the purple one.
Okay. Okay, that's what
So
I
there's,
needed to know.
And then potassium goes.
Yeah.
Do we just leave it for now or what?
so what I want to see it is like things all moving one at a time because it's like steps, right? Like this, then this, then this, then this. So you can use your thing you created to show those steps one at a time. And then you could take turns like actually saying it and doing it. And then you'll actually remember it.
We have a question.
Yes.
So when this one activates this one to open,
Oh God, I need my thing. I need to know what the colors are because I can't remember what I picked. Okay.
when the sodium voltage one opens,
Yeah.
lets sodium in,
Uh-huh.
opens the next one, does
Yeah.
that one then close?
Yes.
Okay. So do we do close them up? So then that'll close and then open that one, lets it in, and then closes and lets that one in.
Right.
Okay.
And then as soon as the sodium comes in, what's pink? Then the pink one
Yeah, it
opens.
opens
Right.
and then it closes once it opens.
And that lets that out and then closes.
Right. hmm
Okay.
And so then,
And then the pumps pump 2 potassium in and 3 sodium out.
right. So then you know, we have to imagine there's another pump over here
Right.
to get this all back to normal. Yeah, well, you can have them pump through there because, you know, they're all diffusing around, right? So they can go out of there.
Okay.
And these get, wherever they went, they can get pumped back in. All right.
Anyway, I can help you.
Uh-huh.
I have a question.
Yes.
So these are the positive ones.
Yes.
That attaches to the cotton ball, attaches to that,
Mhm.
and then sews it
And then it opens it.
and opens it, and then it goes through,
And so then these come in.
and
And then it goes through all the way down here,
Uh-huh.
which opens this.
Opens the yellow ones.
The yellow ones, not
Right.
the pink ones.
Yeah, because the yellow's the sodium channel.
and then
So then they open and they let more of these in. Right?
So when do the pink ones open?
Now, as soon as the sodium ions come in, then the pink ones open.
to let out the pink ones are the potassium.
Uh-huh.
Yeah, those come in.
Well, they let the potassium out.
Oh, they let potassium out.
Hmm.
So now I'm confused. I thought it was over here where the potassium went in and
The potassium is going to go back in. So all this that you're doing does happen the same all down here. So it goes, these open, then the sodium one's open, then the potassium one's open, potassium leaves. And then these, make the next ones open,
and those let some more in,
right?
and then this
So
one
that
lets
more
some
in,
out, right?
right? And then you'd have this pink one open then too, right?
It's like a whole process.
Right, and it just happens going down like the same thing over and over.
It's like the gangster kitchen. Have you ever heard those recipes?
What, y'all putting them up already?
Oh, I thought we were done.
Did everybody do it?
Yeah, we all understood it.
Did everybody like move stuff though?
We were moving, yeah.
I don't believe ya.
Channels.
No, the ligand gate's open. And the neurotransmitter binds. The ligand's open, all the sodium goes in. It opened the ligand gate. No, the neurotransmitter opens the ligand. Is that what you said?
Cool. So how y'all doing?
We're on the last step, the sodium potassium pump. Sodium ions out.
Okay, and which one's your potassium pump? What color is that one?
The yellow Play-Doh, the Play-Doh.
Oh, that's the potassium ions, yeah. But who's the pump?
Oh, the blue one.
Uh-huh. Okay, so you're having the blue ones like move stuff then?
Yeah, so the blue ones move the potassium out. No.
In?
It's potassium in and sodium out.
Right. So, you'd be like taking it in, taking the sodium out through there, right? But not just through the membrane, it's actually leaving through that channel.
Oh, okay.
See what I mean? Because the membrane doesn't let things just go. It has to go through these special channels. So Angela, why don't y'all come over here and move everything then? Because then you'll really know it. All right, so how y'all doing?
I think we already finished with the whole thing right here when the channel is open.
Uh-huh.
Sodium goes in, and then both that together, the potassium and sodium, around the whole
Yeah.
system. And then finally, when you reach the high positivity, then the sodium comes out and the potassium goes in. That's right? No.
Well,
For the pump.
your potassium still needs to leave though, right?
Yep.
Oh, that's with the potassium pump.
Right.
Oh, okay. Channels.
Potassium channels. So like this, to me, you're like halfway, right? So like you let in all your sodium, and then right after the sodium started coming in, what color is it? The pink one's open. How are you showing how these open?
We're just like
We try to move.
Oh, okay. So then they open, and these leave through there,
okay.
right? And then your pump, which is a blue one, then this guy's gonna open and start pumping these in and pumping these out. So then eventually we'll get back to normal.
Okay. So what's the point of the potassium leaving? Just to make it more negative inside the cell?
Yes. And that's actually the reason the cell keeps so many potassiums in there, is so that they have something that can leave and make it negative really fast.
Okay, and then it just gets more negative as the sodium potassium pump works.
Right.
And then, can you explain repolarization a little bit better? Is it that process?
So when the sodium came in here, we became depolarized, so we're positive now. And then when these potassium channels open, that's positive ions leaving, which is repolarizing. And then, the sodium potassium pump helps a little bit too, because it does 2 of these and 3 sodiums. All right, so did everybody take a turn moving all that stuff around? I don't believe it at all. I know it's the two of them moving stuff.
I like the way that he explained it.
research
Because this is like, it
Yeah.
confused me more. Like, this confused me more a little bit. So I
Well,
like the way that he explained it to me.
It opens.
that's because to do this, you have to really know it.
This sodium goes in. Potassium.
Come out.
Okay, that's kind of the start.
And the, when you forgot that one.
What was the first thing that happened down in the dendrite down there?
The
Comes in, right?
Yeah, so the neurotransmitter binds the purple one, it opens.
So it lets sodium in,
Uh-huh.
and you build up a potential, right?
Yeah, potassium doesn't come out yet.
oh,
All the potassium's inside right now.
You build up a potential, and then these are all here too. So eventually, at the voltage, these are voltage gated, so potential builds. These will start to open, right?
Uh-huh.
Sodium comes in, and then which one?
Well, let's see the sodium come in. Did y'all move the sodium in?
I don't even know which type cleaners which ones I'm looking at.
I know.
It's confusing me.
Yellow.
Yes, okay. So sodium's flowing still through yellow, right?
I know. I
So
gave y'all.
here, the leak, so this is the potassium's gonna start coming out eventually.
Uh-huh.
So as this builds, and then it's gonna travel.
So the sodium ions are going down to the other yellow ones.
I don't know why these are here. We moved them. We moved them on down.
Yeah, yeah.
Yep. So then this starts building up, causing the potential to change, right?
And then they
So
should
then-
open.
Then it opens, yeah. And these start coming. Well, these are already outside the cell. Coming in. We're limited here. And
I
then-
know.
so then
Well,
it's-
that's because Andrew's got all the popcorn. He's like gonna eat it or something over there.
You wanted to eat the
gonna-
Play-Doh.
he's gonna
Yeah.
eat the Play-Doh.
Oh
Come
no.
on, give us some crumbs.
Oh no.
Ew, gross. There's some of my hair in there too.
Oh, nasty.
That's so nasty.
Cool. All right. So highly suggest if you aren't totally sure how that all works, I'm sure you can find some materials at home to do
I took a
this.
picture of it. So I'll do it mentally.
No, I want you to do it for real. Get some Play-Doh or draw pictures, something. And you can just put, you can put all this stuff back up here. Put all these colors here.